My name is Praneeth. I'm a first-year graduate student in the Department of Brain and Cognitive Sciences at MIT. I took this image when I was working in Korea as part of a summer school in the laboratory of Dr. George Augustine. We took this image while we were studying circuitry in a part of the brain called the cerebellum. So first, let me walk you through where this picture is in terms of inside the brain. So we were using a mouse. So if this is a top-down view, if you're looking at the mouse print from above, so that's the cerebral cortex, and the two lobes behind is the cerebellum. So we took a slice of the cerebellum like that, and when you magnify that a little bit, you're going to see folds like this. So if I take a small part of one folio, what I'm going to see is three layers. So in the middle layer, there are what is known as Purkinje cells in an ordered manner, and this is what you see in red in the picture because we fill them using a dye. And the green cells that you see where the Purkinje cells make connections in the picture are known as interneurons. So what we were trying to do is study how these Purkinje cells connect to the interneurons and how this leads to information processing inside the cerebellum. So this particular brain slice was taken from a genetically engineered mouse. So this mouse was expressing two proteins which are not normally expressed in a normal mouse. So one of them is called channel rhodopsin and the other one is a green fluorescent protein. So the green fluorescent protein makes the interneurons look green in the slice in the image, and the channel rhodopsin is a light activated ion channel. So, and this is what we were using to study how these interneurons connect to the red Purkinje cells. We focused laser beams using the microscope onto the slice, and because of channel rhodopsin, these laser beams were able to activate these interneurons. And while they were activating these neurons, we were recording electrical signals from the red cells, the Purkinje cells. And this is how we were trying to map the cerebellar circuitry. It's quite hard to get such nice images and to maintain the slice long enough to do two separate recordings side by side so that I can highlight two cells right next to each other without their trees completely overlapping. So it had a certain aesthetic appeal, and it kind of highlights some of how beautiful it is inside the brain.